Good morning to all. Welcome to the Diamond Group of Institutions NEET and K-CET online classes. Today's class, biomolecules, continuation of the previous one. So some of the important and the model questions of biomolecules. First one, molecules having group of opposite polarity. So here they're asking molecules having groups of opposite polarity. So which of the molecules are they contain as the opposite polarity, that is uh, positive charge and the negative charge. So such type of ions are we call as options are they given as zwitter ions, anions, cations and negative ions. So which of the molecules are they contain as a both of the positive charge and the negative charge? That is anionic nature and cationic nature are present. Such type of the ions or such type of the molecules are we call as a zwitter ions. Direct question only. So here some of the molecules at the time of formation of conversion of amino acids or formation of the amino acids. So these are the amino acids conversion of the nitrates into the amino acid formation. Otherwise, ammonium ions are converted into the amino acid formation. Some of the intermediate stages will be noted. That intermediate stages are they contain both positive charge and negative charge into the single molecule. That is called as a zwitter ion. And next question, according to induced fit theory of the enzyme action. Here, enzyme actions are two types. Those are lot and T hypothesis and induced fit hypothesis. So now the difference between lot and T hypothesis and induced fit hypothesis. So if you can imagine about the lot and T hypothesis. So here lot so this is the lot and we are not inserting a specific T. So by the keeping of the specific T into a lot, the lot will be don't open. So here latent T model hypothesis it is. So by the binding of substrates into the enzyme, so that enzyme does not change its structure. So you can imagine about the T contain as a different molds. So in these are the most particular T we are going to insert it into our lot. It's contained as a different mold and T also contained as a different mold. In between the lot and T, that mold interaction it was positive. That lot will be don't open. So here the lot is considered as an enzyme. So when we are don't inserting as a T in the lot. So these are the moles and the T moles will be don't match, the T will be don't open. So here any conformational changes are not takes place into the lot and T. So that is called as a lot and T hypothesis. So substrates always bind into the active site of the enzyme and these are the active site of the enzymes are does not change any conformationally after the inserting of substrate, after the inserting of the T into the lot. That's called as lot and T hypothesis. And question it is asking about the induced fit hypothesis. Means of induced fit hypothesis. In this induced fit hypothesis proposed by Postland, according to him, so when you are not inserting as a T, so when you are don't, you can imagine this is the enzyme and this is the substrate. So actually the enzyme active site structure is different and the substrate structure is different. Then also in this of the induced fit model, so that substrate will be drawn to bind. Substrate will be drawn to bind in the active site of the enzyme. By the binding of the substrates into the active site of the enzyme, after that these substrates will be drawn to induce the conformational changes of the enzyme. So now these are the enzyme active site structure, otherwise enzyme structure will be not modified and substrates will be drawn to bind very tightly on the active site of enzyme. So that is we call as an induced fit model. If the substrate is bind according to the structure of substrate, that enzyme active site conformational changes are take place. That is called as an induced fit model. But in a latent T model, So such type of conformational changes are not takes place. Those are permanent. So that is a latent T model and these are the induced fit model. So question asking about the induced fit model. According to induced fit theory of enzyme action, substrate induce the conformational change in a enzyme. Substrate change its shape after binding and transformational change takes place in a substrate. There is no transformational change in a enzyme. So transformational change is not take place means that is we call as a latent T model. Latent T model. And conformational changes takes place in a substrate. See now here. Here the structure of the substrate is there. After the binding of the substrate into the active site of enzyme, it's having same shape. So option C is incorrect. And substrate changes its shape after binding. Substrate changes its shape after binding. Here also they are asking now after the binding of the substrate does not change its structures. It can change or it can modify. So option A, substrate is induced conformational change in an enzyme. So after the binding of these substrate, by the tightly binding of the substrate into the active site of enzyme, this enzyme active site will be modified because of the presence of substrate. So answer it is A. Substrate is induces the conformational change in a active site of enzyme. And the next question here, they given as one equation Sg plus S that gives rise to S plus Sg. So here we are know the types of the enzymes. Out of the different types of the enzymes, so based by the seeing of this equation, we have to identify it is belongs into the which type. See now, they given as a different types of the enzyme names here: dehydrogenases, transferases, hydrolysis, and the lyases. So first you see about the lyases, means of lyases. So here this type of the lyases are means what's the mean of lyases. So here the lyases functions are then formation of the molecules by the breakdown of double bonds or adding of the double bonds from the substrate. So products are there formed. So these are the substrates are that removed out from by the breakdown of the double bonds or formation of the double bonds. Those are called as lyases. And hydrolysis. Hydrolysis. Here the molecules are then formed by the hydrolysis process, by the involvement of the water. These are the molecules that is we are also called as breakdown concept will be don't take place into the hydrolysis. And coming to the transferases. Transferases represented as transferring of the molecules from the one substrate into the other substrate. Transferring of the group of molecules from the one substrate into the other substrate, that is called as transferases. And dehydrogenase. It is also the breakdown process only, but at the time of the breakdown, removal of water is take place. Water is removed out, that is called as a dehydrogenase. Water is involved, that is called as hydrolysis. And lyases represent as formation or the breakdown of the double bonds that take place, and transferases transferring of the molecules. So now you see S and G, these are the molecules, and here you are going to add it into the S. So now these are the S molecules, they can transfer the molecules from the S from here to here. So they can form into the complex molecules as a S plus S and G. So here one molecules are they can transfer from the one group into the other group. Their specialized enzymes are involved. That enzyme is called as a transferases. So now the answer is transferases, and here the transferase enzyme is involved. And the next question, select the incorrect statement. So we have to identify the incorrect statement in the given options. Option A, collagen is the most abundant protein in the whole biosphere, and rubistro is the most abundant protein in a animal world. See now here, collagen is the most abundant protein in a animal world. They given as a biosphere, and rubistro is the most abundant protein in a plant world. So here they given as a indirectly means reverse direction. So collagen is the most abundant protein in animal world, and rubistro is the most abundant protein in a plant world. That is the correct statement. And option B, proteins are heteropolymers made of amino acids. Heteropolymers means different types of. So proteins are the polymer of amino acids, group of the amino acids. These are the group of amino acids are different types. So different types of the amino acids means we call as hetero only. So polymer means many. Many different types of the amino acids are they combined together that can form into a polypeptide chain that is only we call as a protein. So those are the polymer of that is the proteins are the heteropolymer of amino acids. And ribozymes are nucleic acids with catalyzed power with catalytic power. Ribozymes are the nucleic acids only. They have catalytic power. Catalytic power means they can act as a proteins. They can have a chance. They have a changes. They have a energy to break down of the molecules. And option D, proteins, nucleic acids, and polysaccharides are the only three types of macromolecules are found in living system. They are mentioning as a macromolecules. Macromolecules means these are the components or molecule size will be present as more than 800 Daltons. Which of the molecules are they having more than 800 Daltons? Those all are with all as macromolecules only. So in this all of the living system, in the living system, macromolecules are these three types only. Micromolecules are represented as many. They're mentioning as a macromolecules only. So that's why option D is the correct. Option C also correct and option B also correct. In the option A, the statement they given as a reverse collagen is the most abundant protein in the animal world and but rubistro is the most abundant protein in a plant, what it should be. Rubistro is the most abundant protein in a living biosphere. So in that statement is A. The next question, which is used to staining of lipids? Which is used to staining of the lipids? Staining means coloring. To the identification of the lipids, which reagents are we are going to use? They are given as rhodamine, iodine, ethidium bromide, and Sudan red. You know about that ethidium bromide. After the agarose-gel electrophoresis, to staining of the DNA, we are going to add it into the ethidium bromide that can form into orange color bands on the agarose gel. So that is a nature of ethidium bromide. We are going to use into the detecting of the DNA and DNA bands on the agarose gel. And iodine. Iodine it is used to the conformation for starch, for detecting of starch. And rhodamine. Rhodamine is the molecules we are going to use into the determine the flow of water. We are going to use it to determine the flow of water. So by the using of this rhodamine molecules, the flow of water we are going to easily identify by rhodamine and remain as a Sudan red. Sudan red is used into the detecting the lipids. By adding of the Sudan red, lipids are then absorbed and it's becoming as a red in color. So answer is D. Next question. Which one represents the structural formula of basic amino acids? See now here. Based upon the structures, amino acids are classified into three types. Those are acidic, basic, and neutral amino acids. So how we are going to identify it as acidic amino acids, basic amino acids, and the neutral amino acids based upon the structure. Acidic amino acids. In a structure of the amino acids, more number of carboxylic groups are there present. Which of the amino acids contains more number of carboxylic groups? Such type of the amino acids are we call as acidic amino acids. And which of the amino acids are they contains more number of amine groups? Such type of the amino acids are we call as a basic amino acids. And neutral amino acids are equal number of the carboxylic groups and the amine groups are there present. So now you see the option A, 1 and 2 carboxylic groups are there. So if the carboxylic groups are more, such type of the amino acids are we call as acidic. And option B, number of the carboxylic acids. Number of the carboxylic acids are here 1. So now these are the B amino acid we are going to represent as a neutral. And option C, here also 1 carboxylic group and one amine group it was present. So that's why this is also we are going to represent as a neutral amino acid. And option D, see now Here the one amine group and here the one amine group, here one carboxyl group only present. Such type of the amino acids are we are called as a basic amino acid. So out of the given four options A, B, C and D, so A is a acidic amino acid, B and C is a neutral amino acids and D is a basic amino acid. So question also they are asked, we have to identify the basic amino acid. The basic amino acid is D. And the next question, which biomolecule is directly matched? Which biomolecule is directly matched? Listing, they given as a phosphorylated glyceride found in cell membrane. Yes, listing is a phosphorylated glyceride we are seeing a cell membrane of the all living organisms. The cell membranes are we call as a plasma membrane. And option B, palmitic acid, an unsaturated fatty acid with 18 carbons. Palmitic acid is an unsaturated fatty acid. It's not contained as 18, it's contained as a 16. 16 carbons. 16 carbon atoms are there present. If the 18 carbon atoms are there present, means we call as it's a stearic acid. And adenylic acid. Adenylic acid means it's a nucleotide. So adenylic, adenine is a nitrogen base. It is associated into the phosphate molecule and a sugar molecule, that is adenylic. So adenosine with glucose phosphate molecules. Adenosine means in adenosine it's a nucleoside. See now, this is the adenosine nucleoside molecule, but these are the nucleoside with phosphate molecule we can call as a that is nucleotide with phosphate. Adenosine with phosphate, it should be becoming as a adenylic acid. So now the phosphate molecules are derived as a glucose phosphate. Here they are mentioning as a glucose. So in that statement it is option B, option C. And alanine amino acid. Alanine is an amino acid. It's contained as one amine group and acid group anywhere in a molecule. Alanine is an amino acid. It's contained as one carboxyl group, one amine group, and it's also contained as a one methyl group. So alanine is an amino acid. It's contained as one amine group and acid group anywhere in a molecule. It is also contained as a methyl group in a amino acid. So option B, C, D. So D they given as alanine amino acid. It's contained as a methyl group, and adenylic acid, nucleoside with a phosphate molecule, it's formed as a nucleotide. That nucleotide as adenylic, but here they given as a glucose phosphate. So and option D, palmitic acid. Palmitic acid is an unsaturated fatty acid only. It's contained as a 16 carbon, but they given as a 18 carbons. So B, C, D are the incorrect statement. Option A, lectin is a phosphorylated glyceride found in a cell membrane. That statement is A. And up next question, choose the correct combination. Choose the correct combination. Fatty acid. Now only we are going to discuss fatty acids. Fatty acids, palmitic acid, and phospholipids. So these are the plasma membrane or cell membranes are they contain as a plasma membrane or cell membranes are they contain as a specialized phosphorylated glyceride, that is we call as a lectin. And aromatic amino acid. Aromatic amino acid and acidic amino acid. Acidic amino acids are they contain as a more number of carboxylic group, and remaining amino acid is the aromatic amino acid as tryptophan. So now the correct combination, fatty acids are they given. Fatty acids are example for the palmitic acid. Phospholipids are examples as a lectin. Aromatic amino acids are example for the tryptophan. Acidic amino acids are examples for the glutamic acid, because they contain as more number of carboxylic groups. So the correct combination is B, A4, B3, C2, D1. The next question, which of the following is not a macromolecule? So here biomolecules are based upon its weight. Those are classified into two types, bio micromolecules and bio macromolecules. So here these bio micromolecules are they contain as a less than 800 Daltons. Less than 800 Daltons weight it was. And biomacromolecules are that they are represented as more than 800 Daltons weight it was present, though it is called as a biomacromolecules. So now here proteins, nucleic acids, and polysaccharides are the biomacromolecules, and also we have seen all of the when we done the filtration. These are the biomicromolecules. All are they are present in a test tube, filter, and biomacromolecules are we are seeing in a funnel. So here exemption for the biomacromolecules, which are the molecules are they present in a test tube, that is called as a filtrate. It's also called as a supernatant. These are the supernatant. All are their presence as a micromolecules, and which are the molecules are they present in a funnel. Those are called as biomacromolecules because they are having a large in size. But here one of the molecule its molecular weight is less than 800 Daltons, but it was available or it was present in a funnel because its molecular weight is less only. But here these are the molecules are they present in a cell membranes and cell debris, so that's why they are unable to transfer to the filter paper. So that's why these one of the micromolecule only they are available or they are present in a funnel. So that is called as a lipids. Lipids are the biomicromolecules, but they are available in a funnel. So, which of the following is not considered as a macromolecule? So, proteins, so nucleic acids, and polysaccharides are the macromolecules. They contain as more than 800 Daltons weight. But lipids are micromolecules only, but they are available in a funnel. Answer is D. Next question. Feedback inhibition of enzyme is due to. See now here, it is also the one type of the inhibition of enzyme action. So that in enzyme actions we are going to discuss as a one type of the competitive inhibition and non-competitive inhibition and feedback inhibition. So in that feedback inhibition means that in a multi-step reaction, end products in a multi-step reaction, end products binds the first enzyme. That is we call as a feedback inhibition. Or in a multi-step reaction, end products are that can block the first enzyme. That is called as a feedback inhibition. And these are the feedback inhibition. Option A, they given as substrate accumulation, structural similarity between the substrate and the inhibitor, end products, and any one of the above. So feedback inhibition that can play a key role of the end product. These are the end products are that can bind into the first step of the enzyme. And option B, they given as structural similarity between the substrate and the inhibitor. So structural similarity presence in between the substrate and the inhibitor we are called as it's a competitive inhibition. And substrate accumulation. Substrate accumulation will not consider. So that's why for the feedback inhibition they can play a key role as a end products. For that feedback inhibition, we can take an example as a glycolysis. In a glycolysis is a 10 steps reaction. Glucose 6-phosphate will be known to blocked by the hexokinase enzyme. Hexokinase enzyme, it can blocked by the glucose 6-phosphate. So that is a example for feedback inhibition, and the answer is the end products. And the next question, from the following groups, select the one which has only secondary metabolites. You know that primary metabolites and secondary metabolites. So secondary metabolites are the molecules are they synthesized after the primary metabolites. So now in this we have to identify only the secondary metabolites in the given options. So option A, arbin, cellulose, arginine, and tyrosine. In these are the option A, arbin one only the secondary metabolite. Cellulose is a carbohydrate, and the argine and tyrosine those are the amino acids are the primary metabolites. And glycine, drums in the option B, glycine is the amino acid, drums are the secondary metabolite, serine and diterpenes are the secondary metabolites. So serine is the amino acid and glycine is the amino acid. Gums and diterpenes are secondary metabolites. And option C, carotenoids are the secondary metabolites. Phenylalanine is the amino acid. Curcumin as a secondary metabolite, and rubber is a secondary metabolite. And option D, quantavalin A is a secondary metabolite, morphine is a secondary metabolite, and codeine is a secondary metabolite, and vinblastine is a secondary metabolite. So out of the given four options based upon elimination method, option D they contain as a all or four secondary metabolites only. So only secondary metabolite contained as the option is D. The next question, double helical model of the DNA was given by. So double helical structure of the DNA was given by option A is a Watson and Crick. And what about the remaining options? Knoll and Rusta they invented the electron microscope. Messelson and Stolz, these are two scientists they can propose the conformation test for DNA replication. And M.L. Fisher they can propose the lock and key model. So Watson and Crick, these are the two scientists that can propose they can explain the DNA structure. Double helical structure was explained or given by the Watson and Crick. Answer A. And next question, which is mismatched? So agar is a polymer of glucose and sulfur containing as a carbohydrate. Agar it was extracted from the red algae. It's a polymer of galactons, or we are also called as galactose. It's not a polymer of glucose. They're given as a polymer of glucose, but instead of the glucose, galactose it should be present. And option B, chitin, polymer of glucosamine. Yes, chitin is a cell wall of fungi, and these are the chitins and made up of by polymer of glucosamine, that is we are also called as NAG, N-acetyl glucosamine. And option C, lipopolysaccharides. So lipopolysaccharides are the complex of lipids and polysaccharides. So we have seen above these are the lipopolysaccharides in cell membranes and also the cytoplasm of a cell. So complex of lipids and the polysaccharides. And glycogen. Glycogen is a reserved food metal in animals, and it is made up of by the polymer of glucose. Many number of glucose molecules are connected together with the glycoside bond with 1,4 linkage. So that is we are going to represent as a glycogen. So here out of the four options, mismatched options are. So glycogen is a polymer of glucose. Lipopolysaccharides are the polymer of lipids and the polysaccharides. Chitin is cell wall made up of by NAD, N-acetyl glucosamine. And agar is a polymer of galactose, but they given as a glucose. So mismatched option is A. And the next question, cane sugar hydrolysis to form. So cane sugar, that is sugar cane, will be drawn to hydrolyzed by the adding of water. It will be drawn to becoming as a. Option A they given as glucose and fructose. Option B glucose and glucose. Option C glucose galactose, and option D glucose and maltose. So cane sugar we are going to done the hydrolysis. It will be going to form as a one molecule of glucose and one molecule of fructose. Answer is